Hi everyone, today we are going to explain about a paper titled Chromosomal DNA Fingerprinting, a New Species and Strain Identification Applicable Pathogens. This video is prepared by WARDAH, SABIRIN and MUHRA. What do you know about DNA probe? A DNA probe is a piece of DNA which contains a nucleotide sequence specific for the gene or chromosomal region of interest. Application of DNA probe technology, it offers the possibility of faster and cheaper identification of pathogens, detection of strains with non-virulence factors that have antibiotic resistance, detection of infectious agents in clinical specimens and contaminated food. For microbial identification, there are currently different diagnostic tools that is available to detect different types of bacteria such as Mycobacterium tuberculosis, E. coli, and Mycoplasma pneumonia. Therefore, we can use DNA probe technology for providing DNA fingerprinting for identification because it's highly sensitive to minor genomic variation in nucleotide sequences, and also closely related bacteria can be identified and characterized very accurately. The aim of this study is to describe the uses of chromosomal DNA fingerprint in clinical and public health microbiology. This includes related methodology, application to species and strain identification, ability for bacterial typing. DNA homology measurement between genomes are not sensitive enough to detect the small variation within species revealed by the serotyping, bacteriophage typing, and resistotyping and biotyping. Therefore, we can use DNA probe technology such as whole cell protein electrophoretic profiling, chromosomal restriction enzyme analysis, bacterial restriction endonuclease digest analysis. Bacterial restriction endonuclease digest analysis can detect minor genomic differences between microorganisms in a very sensitive and direct way. Chromosomal probe hybridization pattern. Nucleic acid probes are used in Southern blotting hybridization analysis to highlight specific DNA restriction site heterogeneities. This detects strain differences both within and between species. Types of probes. The difference between short probes and long probes.
Applications of chromosomal DNA probe fingerprint. They have conducted studies on number of microbial groups via chromosomal DNA probe fingerprint to differentiate between a allied species or strain from the same species. In 1986, Griment and Griment used 32 P-labeled E. coli, 16 plus 23S ribosomal RNA probe to discover ribosomal RNA gene in C40 of different gram-positive and gram-negative species such as fingerprint of mycoplasmas and allied genera. They found that they can use plasmide PMC5 as a probe, but because of the high conserved nature of prokaryotes ribosomal RNA gene, they found that they have to hybridize it with DNA containing RNA gene.
ACOR1 restriction enzyme was used to distinguish between 10 non-typable isolates of Haemophilus influenza, and they found that all bands were heterozygous except those who were epidemiologically related strains. They had the same identical pattern. In Salmonella, cloned chromosomal DNA was used to differentiate between 3 Salmonella serotypes. The probes used were successfully differentiating between 3 of them by revealing unique fingerprint patterns. BAMH1 and ACOR1 sites were used within ribosomal RNA genes to distinguish between 2 different mycobacteria, and they found that the sequence of ribosomal RNA genes were significantly different in the 2 species. Both Flagella and Campylobacteria also use the same method. Therefore, DNA printing with the probes can be used as the identification method in epidemiological studies. DNA fingerprints of other microbial genera. Plasma blood hybridization analysis could be used to distinguish both between different species and between strains within species. Advantages of DNA fingerprinting by proper hybridization: applicable to any microorganism, reproducible, wide range of restrictions that are available, commercially available ribosomal RNA can be used, highly sensitive. Disadvantage: time-consuming, detected locally by the probe may not be present in the genome. In conclusion, this soluble method is important to distinguish between related species and help in detecting variation among strains within species. That's all from us. Thank you.